accomplishments of our speaker today, but I'm going to forego it just for a quick anecdote. It was probably 10 years ago. I sat in grand rounds at the University of Florida, and I would always see these introductions and wonder, what's going on with this? Like, who is this person introducing this other person? What's going on here? And it's really nice to be here to introduce our speaker today, because I just probably have hours of things to say about how wonderful this person is. This is Dr. Zabetian. He is a professor at the University of Washington and at the Veterans Affairs, or the VA of Puget Sound. He is one of my mentors. He is a good friend. He has so many clinical trials under his belt. He's a PI in so many studies, and he is the master of genetics in Parkinson's disease. So I'm so very, very excited to have him speak to us today. I think that's just what I'm going to keep it at. I'm happy to be here, but I'm even happier that he is going to be our speaker. So with no further ado, Dr. Cyrus Zabetian. Thanks.
Wow, that was quite an introduction. And I must say that you might notice a difference between the photo that was just shown and me now, because I actually think that was back when I was just finishing residency. So thanks very much. Appreciate you all being here. I'm going to start sharing my screen. And in a minute here. Okay, can you all see my slides?
Looks great.
Okay. So today I'm going to talk about the genetics of Parkinson's disease. And I hope to achieve four objectives today. One is to have you all recognize that genetic risk for Parkinson's disease occurs along a continuum. I'm going to summarize some of the causal and susceptibility genes that have been found to date, and talk about a little bit about what causal versus susceptibility gene means. I'm going to review some appropriate uses for clinical genetic testing, and then I'm going to close by talking about how genetic data might be used to influence our decisions about clinical trials and ultimately precision medicine. So just as by way of background, and this is somewhat arbitrary, but we tend to divide genes for all diseases, Parkinson's disease or any others, into causal genes, where if you have a variant in that gene, you're highly likely to develop the disease. And then risk or susceptibility genes, these are variants that are found in GWASs. And here, there's a very small effect on disease risk, but the variants tend to be very frequent, and the vast majority of carriers will never go on to develop the disease. So if you put this in graphic form, you can see that on the X and Y axis are disease risk and the frequency of the variants. And so in the upper left-hand corner, you have causal genes. These are genes where the mutations in these genes are very rare, but they have a big effect on gene function, and they have high penetrance. So the majority of patients are going to develop the disease if they live to be elderly. And on the other end of the spectrum are these much more common variants that are typically found in GWASs. And they are, again, these are things that have odds ratios that are generally very small, somewhere in the order of 1.1 to 1.3. And then with Parkinson's disease, this is not always the case with other diseases, but in Parkinson's disease, you have some things in the middle. So there's a variant here, LRRK2G2019S, which many of you probably know about, that has reduced penetrance. So its penetrance is probably in the order of 30 or 40%, and then GBA. And so I do want to point out one thing, and that's that there are some genes where different variants can cross categories. So for instance, with the LRRK2 gene, there are rare variants in this particular codon that have very high penetrance. Then there's LRRK2G2019S, which has substantially reduced penetrance. And then there are LRRK2 polymorphisms that occur, with minor allele frequencies in the 5, 10, 15, 20, 30% range that have very small effects on disease. Again, and they, not surprisingly, these variants also have varying degrees of effect on protein function. So big effects, more modest effects, and much smaller effects. And so if we look at how causal genes are generally found, if you think about the population in North America, about 20, sometimes up to 25, 30% of people will report a relative with Parkinson's disease. So we'll classify them as familial. The rest of them don't report any family history of the disease. And then there's a very small subset of people who come from a family that's large enough and shows clear Mendelian inheritance, where there's multiple affected members of the family, where you can really see that something looks autosomal recessive, autosomal dominant, X-linked, et cetera. And it's that small subset of larger families where much of the gene discovery for causal genes has occurred. So typically, what ends up happening is those families are recruited and enrolled, and in the old days, you do linkage analysis, which would show you regions that were oftentimes in megabases across that you honed in on. You did sequence analysis and various other sorts of things, and eventually, if you were fortunate, you'd find mutation and show that that was the causal gene for your disease. That's changed a lot with the advent of next generation sequencing, and I'll talk about that later in the talk. So the first causal gene in Parkinson's disease was identified in 1997. This was an Italian kindred, the Contursi kindred, where a mutation, a point mutation, missense mutation, in this SNCA gene that encodes alpha-synuclein was discovered to segregate with disease in this dominant fashion. Had a very high penetrance, and the age of onset was typically between 30 and 50 years of age. That same year, it was shown that the primary protein in Lewy bodies was alpha-synuclein, the protein that's encoded by SNCA. And then subsequently, that was a point mutation we were talking about, but subsequently it was shown that duplications and triplications of the normal wild type gene that caused you to have normal protein in terms of its sequence, but elevated levels of the protein overall were sufficient to cause Parkinson's disease. And you could see that there was increased alpha-synuclein in biofluids, including blood, CSF, and brain. And if you looked at the penetrance rates, if you had a triplication, so 3 copies, your penetrance by age 50 was almost 100%. Whereas with duplications or point mutations, it was only about 60 to 70% at age 50. But eventually, in later years, it still had very high penetrance. And this sort of shows a dose effect response curve here. There have been, by now, there are at least a dozen causal genes that have been discovered and reported for Parkinson's disease. I highlight a few here. So SNCA, which we just talked about, and LRRK2 are the most common ones amongst the dominant forms of the disease. Parkin, PINK1, and DJ1 are the most common by far amongst patients that have autosomal recessive or juvenile autosomal recessive Parkinson's disease. And then there's a gene, RAB39B, which we co-discovered a decade ago, which is actually X-linked, but quite rare. And if you sum up all of these mutations together and you look at an unselected group of patients in North America, that's patients that you sort of take randomly off the street without regard to age of onset or family history, probably between 1% and 2% of those patients will have a mutation in any of these genes. Now, if they have a family history, that goes up substantially. And then there are a number of genes. This is not fully inclusive, but there are at least 15 to 20 genes where they have the patients have a neurodegenerative form of Parkinsonism, but it usually doesn't present phenotypically as pure PD. It usually presents as atypical Parkinsonism and occasionally can mimic PD fairly closely. And so when you're testing, as I'll talk about later, for Parkinson's disease / Parkinsonism, most panels will include all of these genes, or many of them. So in 2004, the LRRK2 gene was reported by 2 different groups independently, and several mutations in the gene have been shown to cause disease. But the one that I'll focus on is G2019S, and that's simply because the frequency of G2019S is so much higher than all the other mutations in this gene and all other dominant genes combined. And so this gene, this mutation, G2019S, it turns out it has an interesting history. So we published data almost 20 years ago to show that this mutation probably arose in the Middle East or North Africa about 2,000 years ago, and then spread across Europe and then elsewhere in the world. And so if you look at its frequency in today's world, this is the frequency of G2019S amongst patients with Parkinson's disease. So you can see that with this heat map here, that in Israel and North Africa, the frequency amongst PD patients of this mutation is upwards of 25–30%. It's higher in Southern Europe. In the US, in North America, it's probably got a frequency of about 1% or so in patients, unselected patients, very rare in Asia, and somewhere in between in South America. And if you look at carriers of G2019S by age 80, this was a group that looked at, that separated patients of Ashkenazi Jewish ancestry who have a much higher prevalence of the mutation versus everybody else. And penetrance ranged from 25 to 42%. There have been several other studies that have looked at this, and long story short, most of them show that the penetrance rate is no more than 50%. So we talked about causal genes at this end. Now I'm going to talk about GBA. And GBA is an interesting story. So GBA, this gene, encodes the protein glucocerebrosidase. So we've known a lot about GBA for other reasons. So if you have 2 defective copies of this gene, you develop Gaucher disease, which is an autosomal recessive lysosomal storage disorder, where you get manifestations you get hepatosplenomegaly, bone marrow dysfunction, skeletal disease. You can get CNS involvement. It can range from very mild forms of the disease that only become apparent in adulthood or even late adulthood to forms where it affects children or even the fetus in utero and can be very severe. It was reported over 100 years ago. And essentially what happens is you get dysfunction of the glucocerebrosidase enzyme. And this is an enzyme that breaks down glucosylceramide, which is part of the plasma membrane. It's ubiquitous, and its turnover is just a normal part of cellular maintenance and function. If you block it, you get accumulation of this molecule, glucosylceramide. And what ends up happening is you get this accumulation of this substance occurs in macrophages in multiple organs. This is a bone marrow smear, showing one of these Gaucher cells, which is filled with glucosylceramide. So you might ask, well, what does that have to do with Parkinson's disease? So it turns out that if you have one defective copy, if you are a heterozygote, you're at increased risk for Parkinson's disease. The gene has more than 300 mutations that have been reported. Most of them are missense mutations. And they range in severity in terms of how much they affect protein function from mild to severe to null. So those that are null essentially knock out protein function altogether. Those that are mild affected to a much lesser degree. And not surprisingly then, if you have a mild mutation, your increased risk for Parkinson's disease is with odds ratios somewhere in the neighborhood of 3 to 5. If you have null or severe mutations, and this L44P is an example, you have a much higher risk for Parkinson's disease, somewhere in the odds ratios of north of 10. And there's also a polymorphism, E326K, that if you have 2 copies of it, you don't get Gaucher disease. So it's got a very modest effect on gene function, but it does increase your risk for the disease. And in terms of how common these mutations are, so this is these are data from a consortium that I lead, which has now 15 sites, and we screened 1,369 patients. And these mutations that are reported in Gaucher disease cumulatively amongst PD patients occur with a frequency of about 4.4%. This functional polymorphism E326K, about the same frequency. When you add it up, that's 9%. So that's about 1 in 10, 1 in 12 patients with Parkinson's disease will have a variant in one of these genes. Sorry, in the GBA gene. And subsequently, we and other groups have shown that GBA carriers have a more malignant form of the disease. So they have more rapid progression of motor symptoms, and they convert from normal cognition to MCI or dementia more quickly. And this is just data from the study where, interestingly, we showed that faster progression in motor symptoms is mostly in postural stability, so PIGD score. So those features of the disease, rather than resting tremor. So their tremor scores in these patients declined much more slowly. And then the last category are these GWAS hits, these susceptibility genes. And the advantages of GWAS, so prior to the GWAS era, there were candidate gene studies that would look at a handful of genes based on some sort of biological hypothesis about the disease. And these were oftentimes negative and conflicting. And the nice thing about the GWAS era, which started about 14, 15 years ago, was that it covered all genes at once. So it was model-free. It improved, and that improves efficiency and forces you to present and publish all of your results. It definitely lessens publication bias because of that. And for many years, NIH-funded GWAS studies were compelled to make their data publicly available, and still are today. So the first few in 2009 and 2010 were interesting, but I'll just explain. This is called a Manhattan plot, and many of you have seen these in GWASs of a variety of diseases. But basically, the chromosomes, the chromosome position is here on the x-axis, and on the y-axis is a log of the minus 10 of the p-value. And you have to have, because you're doing you're testing many variants, you have to have a very stringent level of statistical, a high statistical threshold to account for all of the multiple testing. And so the bar for calling something significant in GWAS studies is here, usually around somewhere in the order of 1 × 10 to the minus 8th or 5 × 10 to the minus 8th. So this was one of the very first GWASs that was published, and lo and behold, common variants in synuclein were at the top. This is the MAPT gene, which encodes tau and is a key player in tauopathies. And these were the only 2 that reached significance. And you can see that the sample sizes here, somewhere between 15 and 2,000 cases and 4,000 controls. The following year, we were involved in a GWAS study from 4 sites that replicated these data, but also showed that a variant within one of the HLA loci was also associated with PD. But as time went by, people started doing meta-analyses and mega meta-analyses of these GWASs. And when you do that, you start getting more clarity. So fast forward a decade. This is a study by Mike Knowles and Andy Singleton at the NIH. And they looked at 38,000 cases and over 1 million controls. And what you can see here is, this is a Manhattan plot, but it's hard to read because there are so many genes on here. So basically, they confirmed the genes that you see in this kind of the gray boxes were genes we already knew about. So they confirmed them. And these blue boxes were genes that they only saw as significant when they did this large meta-analysis. And that's simply because of power. So a lot of these variants down here have very low odds ratios and have very small risks. And if they have very small risks, you're only going to see them if you have really large samples. Another thing I need to mention is that oftentimes you'll get a hit in a region And sometimes it's clear that it's in a single gene, but there are oftentimes the case that you'll have several genes clustered in an area, and you'll have hits across that area. And you won't know whether it's any one of those single genes until you do more refined studies. And sometimes even then, it's very difficult to sort out which is the susceptibility gene itself. And a question I often get is, okay, so if these GWASs are finding genes that have such a small effect on risk, what's the point? We get it when you talk about causal genes, but what's the point if you're looking at susceptibility genes? And what I would say is that what is interesting and I think is informative is that when you start seeing these genes stack up in particular pathways, in other words, when there's a biological pathway like mitochondrial function or lysosomal function, where you get multiple GWAS hits in different genes, that gives you a clue that that particular pathway is important and underlies the idiopathogenesis of the disease. And that provides clues on targets for future intervention. So if you look at, this is a diagram that was done several years ago by Billingsley and Singleton. And they essentially stack these genes up by where they fall in terms of biological pathways. So you can see you have many genes, including GBA and a number of others, that are involved with lysosomal function. Several that are involved in endocytosis, some that are involved in autophagy, some that are involved in mitochondrial function, some in the immune system. But that gives you a clue as more and more genes pile up in these particular specific cellular pathways that they are important, particularly important for Parkinson's disease. The last thing I'll say on this topic is that all of the data that I showed was really based on people of European origin. And this was a graphic that was published a few years ago that looked at the population of patients in GWAS studies of all different diseases, not just Parkinson's disease, but all different diseases. So you can see that people of European ancestry here in this red color constitute the vast majority of patients that are in GWASs. Here, you can see the actual global representation. So not surprisingly, people of European origin are a modest chunk of the population globally, but they constitute the vast majority of patients in GWAS data. And so that's problematic because the findings that we have in people of European origin, some of those are going to transfer to people of other ancestries, but some of them are not. And so to try to combat that problem, there's a program called the Global Parkinson's Genetics Program, GP2, that's funded by several different sources, but the Brin Foundation is a major funder of this program, and Michael J. Fox Foundation is the organization that administers these grants. And what they're basically doing is trying to pull together resources and fund studies from across the world. They're hoping to enroll 150,000 PD patients, of whom they're hoping to get 40,000 who are non-European ancestry. And this includes patients who were enrolled in studies long ago where their DNA is archived, and patients who are being prospectively recruited. And we were fortunate enough to get funding from this program. And within the VA, I lead this genetics consortium, which we just launched last year, which includes 22 sites. We in Seattle are the coordinating center, and these sites across the country enroll patients. These are all VAs where there's a movement disorder specialist who's the local site investigator. They gather information, they send us blood back, we do all the processing, and then the DNA is genotyped by the NIH. And we are targeting on a 1-to-1 ratio patients who report their race as white, and non-white patients who can be described, who self-report as either non-white or as Hispanic Latino. We're also gathering data on people's military service so that we can understand where they were in time and space and assess them for environmental exposures to look at gene-environment interaction. I'm going to touch briefly on genetic testing. And I think when I'm ordering genetic testing or thinking about it, there's several things that go through my mind. And a lot of these points apply to many diseases, not just neurological diseases, not just Parkinson's disease. So the first thing is, how likely am I to detect the mutation? Well, from the data I just showed you earlier, that depends on several factors. Do you have a family history of the disease? If you don't, you're much less likely to find a mutation in the causal gene. Age at onset. If you have an earlier age of onset, more likely to find something. Ancestry. So if you are, for instance, of Ashkenazi Jewish ancestry or from North Africa, you're much more likely to have a mutation in G2019S. Then there are clinical considerations. So in some cases, genetic testing defines the disease. So for instance, in Huntington's disease, we don't say that you have Huntington's disease unless you test positive when you're tested. That's not the case in PD. Management of the disease itself. So will the genetic results alter your management? Earlier treatment, your choice of drugs? The answer is not yet for Parkinson's disease, but that might not be the case several years from now. What about prognosis? Well, it is true that the rate of progression is higher for some patients. So for instance, people who have GBA mutations tend to have a more rapid decline in both cognition and motor symptoms, whereas LRRK2 carriers tend to have a slower decline. But there's lots of overlap between groups. And so I generally don't make this a major consideration when I'm ordering testing. Risk of conversion for relatives. So that's a major point, right? Because when we're ordering testing for people who already have the disease, they already know they have the disease. But when you're talking about people who are at risk, there are several things to consider. In theory, loss of employment or eligibility of benefits for disability insurance are things to consider. One of the things I think is a major factor is stress, right? Either getting tested or not getting tested. And some people really want to know the answers. They don't like to have uncertainty. The problem, of course, is that even if they test positive for some of these mutations, it doesn't tell them that they're going to get the disease. It just informs them about risk. Family planning, life decisions. Usually when patients are manifesting with Parkinson's disease, they have already had families, but there are occasionally people who have early onset disease where this might affect these decisions. And then genetic counseling, again, something that I think is important in some way, shape, or form. And that's something that's bypassed when you do direct-to-consumer testing. And then eligibility for research trials. I'll talk about this later. So in the early days of testing, there were a limited number of genes. And this is an example of an Athena panel that was available in the early 2000s. And this was all done with Sanger sequencing, which means that you had to sequence each gene, gene by gene. It was very labor intensive. And this is what was available. And then with the advent of next generation sequencing, things were much easier. So whole exome sequencing is commonly done for research purposes and clinical purposes. And now we've moved to whole genome sequencing being available. And there are many companies offer these gene panels. And just to tell you that these panels can be done either one of two ways. First, you can do whole exome sequencing, but mask all the other genes except for the genes of relevance. That's one way to do it. Another is a capture panel when you're only sequencing the specific genes and you don't have data from anything else. The nice thing about this is that if in a couple of years, a new gene or two is discovered, you can go back and ask whoever did the sequencing to unmask those genes and look to see if there are variants in those particular genes. These are just a few panels of many that are available. This is one that's available at the UW. This is one for prevention genetics, and we at the VA have a contract, so this is where we have to go if we're ordering it at the VA. This is a common panel that's done by Invitae. And you can see that they differ not by the main not by the main PD genes, because they all cover LRRK2, synuclein, PIK1, DJ1. But some of the more rare, some of the genes that have rarer mutations in them. And then another option that people can, in some places, can avail themselves to is PD generation. So this is a study that the Parkinson's Foundation launched that screens 7 PD genes. And the advantage of it is they offer genetic counseling, and it's free. And enrollment can be done, and the counseling can be done either virtually or in person. But again, this is not available to everyone, and there's somewhat of a wait. It's the other thing that I've heard. So just something to caution you all about, and this is the case whether you're talking about genetic testing for Parkinson's disease or any other disease. When you order whole exome or whole genome sequencing, you get a lot of information. And when you do that, the core facility that is generating the data has to generate a report and try to annotate each of the variants that they're reporting out as either pathogenic or benign or something in between. And so there are several things that go into this decision. So one is how disruptive is the variant predicted to be in silico? In other words, is it an area of the protein that is highly conserved across species or not? Has it been reported in that particular disease? And if so, does it clearly segregate with disease? So you take a combination of clinical and biological predictions, and you have to come up with ultimately your prediction. And a lot of variants get stuck. So many variants, the majority of them are benign. There are some that are clearly pathogenic and have been reported in multiple families with that disease. But there are a number of variants of unknown significance. So these VUSs are variants where you really don't know whether it's pathogenic or not. And that may seem like, oh, and one other thing is that because we know less about the genetics of a number of diseases in people who are not of European origin, this is a bigger problem amongst other groups, understudied groups, particularly like Latinos and African Americans. You have less information, and so you are more likely to call things that are probably benign variants of unknown significance. And this problem might seem trivial at first, but it isn't to the consumer. So this was a study that looked at, that surveyed parents of 192 children who had diagnostic exome sequencing for a variety of diseases. And the reports were divided into 3 categories. Reports that had positive results where they had a mutation that was deemed pathogenic, those that were negative, and those where there was a variant of unknown significance. And these are the stress levels. These are simply stress levels amongst the parents. And you can see that there, in fact, wasn't a significant difference in the level of stress between parents who were given a positive result and those that were given a result of unknown significance. So just something that I caution people about when they're ordering whole exome sequencing, or particularly whole genome sequencing, where you get a lot of information, and oftentimes you'll get several variants of unknown significance. So lastly, I'm going to talk about precision medicine. And I know that most of you are familiar with the concept, but just to review it quickly, the idea is that for any disease, there's a patient population that has the disease as we define it. But there are subgroups within that disease that have common characteristics. That common characteristic might be that they share a variant in a particular gene, or they have a particular tumor marker, for instance, if we're talking about cancer, or they've been exposed to a particular environmental exposure, and they're out there in the population, and we may have a treatment that would be very effective with one subgroup, but essentially is not effective in any of the others. And to some degree, this has been going on in cancer treatment for many years. So if we were able to deconvolute and identify these subgroups and stratify by these subgroups, then we could be much more efficient. And you can imagine, if we're purely talking about genetics, treatment A would be very effective for one subgroup, and perhaps that might be a treatment that's targeted to GBA, as an example. And then treatment B might be something that targets LRRK2, Parkin, etc. And there may be a group that you never really find a treatment that's going to be specific for them. And when I'm talking about treatments here, I'm talking about things that are disease modifying. So we don't have really disease modifying therapy other than perhaps exercise in Parkinson's disease. Obviously, with other neurologic diseases like MS, that's not the case anymore. And so there have been trials on a couple of these. There have been trials that have targeted mutations in two of the genes that I've talked about. One is LRRK2 G2019S, and they are the idea that with these mutations is that the kinase activity of LRRK2 is overactive, and so a number of companies have been working on LRRK2 inhibitors. This is a compound by Denali, and Biogen has now joined them, that has gone through phase 1 and phase 1b trials. The results were published in Movement Disorders last year. It was shown to have both good target engagement and pathway engagement and to be safe. And now there's a phase 2 study that's been ongoing since 2022, the LUMA study, that's enrolling a larger group of patients, both patients with LRRK2 mutations and patients without. And that's the other thing. It's possible that these therapies that are targeted to a particular gene could, in theory, be helpful for patients who don't have a mutation in that gene. We just don't know yet. GBA is a hot topic, and part of that is simply because of numbers, right? So 1 in 10 to 12 patients are going to have a relevant variant in one of these genes. So that's, many, many, many tens of thousands, if not hundreds of thousands of patients out there. So there are several ways that you can target the pathway. So first, you can target this molecule that's upstream, actually, of glucocerebrosidase. If you inhibit it, the idea is you are decreasing the amount of glucosylceramide that accumulates, assuming that that molecule is what's causing the problem. So there was a study that was launched by Sanofi, the MOVES PD study. That trial, that second phase trial, was completed and did not slow progression. There are a variety of reasons why that might be, but it very well may be that targeting one area of this pathway might not be effective, whereas targeting another might be effective. Just to let you know that you might ask, well, can't you just give them the enzyme itself? And it turns out you can, and that's been done for many years to treat Gaucher disease. The problem is these enzymes don't cross the blood-brain barrier. So most of what's been targeted now are small molecules that go through the blood-brain barrier into the brain. This is another compound that was developed by Lysosomal Therapeutics, and now biotech has taken over. A phase 1 study was completed. This is a glucocerebrosidase activator, so it actually activates the normal copy on the other chromosome. So it's something that would work for people who are heterozygous but not homozygous. And a phase 2 study has just started that goes by the acronym ACTIVATE. And then finally, Ambroxol, which is an old drug that was used as an anti-mucolytic. Turns out that it works as a chaperone that protects beta-glucocerebrosidase from normal degradation, so it keeps it around longer. There have been phase 1 and phase 2 studies that have shown promise, and now there's a phase 3 study that's planned that I don't believe has launched yet. And so those molecules target glucocerebrosidase itself down here. Again, the idea here is that these therapies, by getting at the heart of the problem, are going to change the trajectory here. So if this is the normal aging curve, and let's say this is number of dopamine neurons, the idea is that with disease comes a steeper trajectory where you cross over a certain number of cells, and then you start to develop symptoms here. And here we're hoping that with these molecules that target GBA or LRRK2 to change the slope of that curve, so-called neuroprotection, so that you're delaying onset of the disease to later in life or perhaps beyond forever, essentially. And finally, I'll say this, that there are most people out there are not going to have a mutation in a gene that has large effect. But it turns out that you can take all of that GWAS data and take the effects, cumulative effects of all of those different single nucleotide polymorphisms and consider them together. And so if you do that, you get this normal distribution where you have people at the very end of the spectrum that are at high risk because of a combination of risk alleles at several different genes. And at the other end of the spectrum, you have people that by luck of the draw are at low risk. And so the idea is that it's possible that even if you don't have a mutation in a single gene, if you're in this category of high risk, you might be somebody that might be eligible for clinical trials of molecules that don't particularly target a single gene. But this just shows you that we can use those GWAS data to try to affect our decision making eventually, hopefully, even for people who don't have mutations of causal genes. So in conclusion, I hope I've shown that genetic susceptibility for PD occurs along a continuum. There are at least a dozen genes for monogenic forms of PD that have identified to date, and at least 15 to 20 that result in atypical Parkinsonism. Targeted studies in GWASs have found nearly 80 susceptibility loci. Again, how most of those genes affect protein function and susceptibility to disease are still being worked out. There are a number of factors to consider when you're ordering clinical genetic testing, and that's particularly true as it's become more comprehensive with exomes and genomes available and thus more complex. And ultimately, we hope that genetics might influence our clinical decision-making as the era of precision medicine unfolds. So thank you for your attention. I'd be happy to take questions.
Yay. Thank you so much. I will read some of the questions we have here, and then, of course, I have questions to ask you as well. So let's see. The first question is from Dr. Bonnie Kong, who is your previous resident. She asks, what do you say when a patient or friend asks you if PD is a genetic condition? And in parentheses, she put, in a short answer way, close parentheses.
So I get this question all the time. And I basically say that it's a combination, it's a combination of genes and environment. And for some people, genes play a big role. And for other people, environment plays a big role. And for some, they both play a big role. I never say that PD is or isn't a genetic disease, because it isn't.
Right. Yeah, it's $1 million question. It's hard to answer. There are multiple studies discussing GBA, for example, and deep brain stimulation. What is your take on the effects of deep brain stimulation with patients who have genetically confirmed Parkinson's?
So in general, it doesn't, and I know there was a paper published that suggested that we should all be doing genetic testing on patients and determining whether they have GBA. And my thoughts are that the issue is that these patients, regardless of whether they have DBS or not, are going to progress on average faster across all both non-motor and motor symptoms. But I don't let that influence my decision at the time. And I think that also goes to the question of, well, should we be testing everybody for GBA anyway? And my answer to that question would have been different a few years ago, but now that there are GBA clinical trials, and most of these GBA clinical trials are looking for people that have relatively early disease. So for instance, the BEAL study that I mentioned, you have to be within 7 years of diagnosis to be in that study. So if you wait around, people aren't going to be eligible. So more and more, I'm talking to people about doing testing, not necessarily exomes or genomes, but for a limited panel, for instance, that gets LRRK2 and GBA for that reason. But I don't use it in my clinical decision making when it comes to DBS.
Right. Yeah. And it's, one set of data will come out saying it's against it, and another set of data will come out and say it's for it. So it's a little bit, I think that's variable, as you mentioned. I think that's it for the questions we have. Dr. Kamani asked shortly after I did, first off, he said, "Thank you for the excellent lecture, Cyrus. In a similar manner, any correlation between genetic mutations and DBS efficacy/durability?"
Yeah, so I think the data that are there mimic what we had talked about, which is if you have a more malignant form of the disease, you're going to progress regardless of whether you take whatever symptomatic medications you're on, whether you have DBS or not. The underlying problem isn't changing, so you're going to progress faster. So yeah, the durability is going to be less of medications and of DBS simply because of that. I think, again, if you know that a patient would respond to DBS, but their durability might be shorter, is that going to affect your decision making? For me, probably not. But yeah, I think that's the problem. And likewise, people with LRRK2 G2019S seem to progress more slowly.
Right. I think that's it. Amazing. As always, thank you so much. Hopefully we get to see you again in Copenhagen having weird sandwiches. Oh, one more question from Dr. Doherty. Okay, some of the epilepsy med data suggests perhaps a provocation of Parkinson's-like side effects, if not Parkinsonism. Are gene tests here for residual Parkinson's symptoms off of anti-seizure meds reasonable?
So if somebody has had an anti-epileptic and they are taken off and they have residual signs of Parkinsonism,
I think it's in.
Yeah, that'd be right, Cyrus. So for instance, let's say you have someone on Depakote and they have Depakote shakes that look very Parkinsonism-ish. You take them off of it and the shakiness, etc., still persists. Is that a state where you might want to look into any gene analysis, or just wait and see what happens, I suppose?
So I guess if they have, and this could be the case with neuroleptics as well, right? Somebody develops Parkinsonism that maybe looks a little asymmetric. You're able to take them off the medication and they still are showing some mild symptoms. Some people would argue that that's unmasking a process that's already going along. I mean, I think there are several things you can do. You can wait and watch, and if they progress, then that's problematic, and maybe you start treating them. Some people will get a DAT scan. Some people like synuclein skin biopsies. I think those are all reasonable. I'd probably go with one of those things before I went with genetic testing, unless the person had a family history. I might do both if they have a family history, but I'd probably do one of those other things first. And you're movement people in your own group. I'd be curious to see what Praveen and Amir do, because I know you guys get patients who are in that very same boat and they get sent to you.
Yeah. I mean, it's exactly what you said: DAT scan, SIN1 biopsy, or perhaps just a levodopa trial to see if if there's significant reversibility of symptoms after a levodopa. There's not many other diseases that do that.
Right. Yep. And also, I think it also depends, right, on the patient. So if there's somebody who has schizophrenia and it's a very difficult time coming off, I'm more careful about doing a levodopa trial. And that happens at the VA more so than in other healthcare systems, as opposed to somebody who has epilepsy, let's say.
Right. And the value of the skin biopsies these days is that the DAT scans can be muddled by some of the medications that the patient's taking. So you get a cleaner result, so to speak, by doing the biopsy. And that's another topic of contention.
And actually, along those lines, we work with collaborators at the University of Washington who are doing the synuclein aggregation assays on blood. And they actually have really promising results, and there have been some results published. And I actually think that within a short period of time, we're going to start doing the aggregation assays on blood, and we may not be doing any of these other things. Well, I shouldn't say we'll still be doing DAT scans, but we may not be doing any of these other things.
Right. Yeah, it's promising for sure. I think that's it.
Okay.
Cool. Thank you so much.
Thank you. Been a pleasure. And if you guys have any patients that have really strong family histories, please let me know, because we have studies ongoing for familial Parkinson's disease as well. And I know that Dr. Kamani is very good about referring patients, and I appreciate that. So if anybody else has patients like that, please let me know. Feel free to email me.
Thank you.
All right. Take care.